Hello students, I'm Dr. Arpita De. Welcome to my biology classes. The topic for today's discussion is ultrastructure of peroxisome and glyoxysomes. So before we start, let's first see what are glyoxysomes. Glyoxysomes are a type of microbodies. Microbodies can be subdivided into two categories, two classes. One of them is peroxisomes, the other is glyoxysome. Now, first question that should come in our mind is what is a microbody? Okay, so now let's see what is a microbody. Fine. A microbody is a type of organelle that is found in the cells of plants, protozoa, and animals. Okay, organelles in the microbody family includes peroxisome. So you must remember microbody is not one particular type of an organelle. Instead, it is a family containing different types of cellular organelles, right? So this microbody family include glyoxysomes, peroxisomes, glycosomes, and hydrogenosomes. Fine. Now today we will concentrate on peroxisomes and glyoxysomes. Okay. Now let's take a look at what is endomembrane system. Endomembrane system is a membranous component of the eukaryotic cell. Okay, you know eukaryotic cells and prokaryotic cells. Eukaryotic cells are more advanced. Fine. So endomembrane system is a membranous component of the eukaryotic cell. It includes endoplasmic reticulum, Golgi apparatus, and lysosomes. Now, why did we suddenly bring this endomembrane system over here? We started talking about endomembrane system to ensure that, to show that microbodies do not belong to endomembrane system. Okay, see, endomembrane system includes endoplasmic reticulum, Golgi apparatus, and lysosomes. Fine. It works together to modify, package, and transport lipids and proteins, breaks down toxic chemicals and cellular byproducts. So this is the function of endomembrane system. What does it do? It performs these type of functions. It works together to modify, package, and transport lipids and proteins, and it breaks down toxic chemicals and cellular byproducts. Now see, this is endoplasmic reticulum. You can see endoplasmic reticulum, or ER, as you know, is of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum, why is it rough? Because it has got ribosomes on it. These dots are ribosomes. Fine. This is your Golgi apparatus and this is lysosome. So this is a diagrammatic representation of the endomembrane system. Fine. Now, specialized peroxisome in plants are known as glyoxysome. Okay. They are usually found to occur in fat storage tissue. Specifically, they are found in fat storage tissues. Okay. Now, single unit membrane bound vesicles containing enzymes. These glyoxysomes are unit membrane bound vesicles. Okay. The matrix is finely granular. These are actually the characteristic features of glyoxysome. Okay. These enzymes convert fat to carbohydrates during germination. When is this fat getting converted to carb or carbohydrate? During germination. Fine. Enzymes for beta oxidation of fatty acid and glyoxylate cycles like isocitrate lyase and malate synthase, all these are available here. In glyoxysomes, the fatty acids are hydrolyzed to acetyl coenzyme A by peroxisomal beta oxidation enzymes. Okay, these are the some of the very important points associated with glyoxysome. Okay. Now let's take a look at the eminent scientist who has discovered this. This was discovered by Christian D. Dove. These are single membrane bound, roughly spherical, small, ubiquitous cell organelle found in the cytoplasm. Okay. The diameter is roughly 0.5 micrometer and it is the site for oxidation of fatty acids. Okay. These features are for peroxisomes. Remember, peroxisome, this is very, very important. You should keep this in mind. Peroxisomes do not have their own DNA. Can you name two organelles who has got their own DNA, own genetic material? You have studied about that. One is chloroplast. The other is mitochondria. These two organelles have got their own DNA, but peroxisomes do not have their own DNA. All right. Now see, let's take a look at how does it look. I've made a very simple diagram so that you understand it in a more easier way. Take a look at this. This is the crystalline core. All right. This is encircled or this is bordered by lipid bilayer and then you have the plasma membrane. Fine. Please learn this diagram. It's a very simple one. Peroxisome precursor vesicles are thought to bud from ER. See, endoplasmic reticulum is thought to give rise to peroxisomes. Okay, peroxisome precursor vesicles are thought to bud from endoplasmic reticulum, which may then fuse with one another or pre-existing peroxisomes. Okay, this will bud from the endoplasmic reticulum, and then what will happen to these? They will fuse with one another or the pre-existing peroxisomes. Fine. Since peroxisomes are not created from the endomembrane system, they are bound by a single membrane instead of a double membrane like most organelles. Please keep this point in mind. Peroxisomes are not created from the endomembrane system. Okay. They are bound by unit membrane. They are bound by single membrane instead of a double membrane. Fine. Think of mitochondria. Mitochondria look like this, isn't it? It looked somewhat like this. This one was the outer mitochondrial membrane, and this one was the inner mitochondrial membrane. So they had a double membrane system. But here, these these peroxisomes do not have that, right? They contain urate oxalate crystalline core. This is the crystalline core. Okay. So what is this crystalline core? It is urate oxalate crystalline core, and it contains more than 50 enzymes, contains oxidative enzymes such as catalase, D-amino acid oxidase, and uric acid oxidase. You must be knowing what are the various functions of these oxidative enzymes. Why are they so very important? I'm not discussing the importances of these enzymes over here. Peroxisomes are membrane-bound packets of oxidative enzymes. Okay. In plants, they convert fatty acid to sugar and assist chloroplasts in photorespiration. Photorespiration cannot operate without peroxisomes. You know what is photorespiration? C2 cycle in plant. Fine. In animals, so this is what is happening in plants, and in animals, they protect the cell from its own product of toxic hydrogen peroxide. Okay. You know, H2O2 is highly toxic. H2O2 is very toxic. So let's take an example. White blood cells produce H2O2 to kill bacteria. The oxidative enzymes in peroxisome break down the hydrogen peroxide into water and oxygen. Okay, H2O2 is there. It is broken down into water and oxygen. All right, it is broken down into water and oxygen by hydrogen by peroxidase enzyme. Okay, so it is neutralized. These two components will not cause any harm. They are neutralized. All right. So this is how we come to the end of our lecture. If you have any confusion anywhere, please feel free to get back to me in the comment section. Thank you so very much for attending the class. Have a nice time. Goodbye.